The new 8,000 square foot forensic DNA testing facility is home to 3 disciplines comprised of 20 DNA analysts and technicians. Although each discipline has a distinct function, they're united in their mission of associating DNA evidence recovered from a crime scene with that of a known individual. A wide array of evidence is submitted for testing. The DNA Casework section performs a physical examination of the evidence to locate biological material containing DNA. Depending on the nature of the crime and evidence, additional chemical tests might be performed. Once the biological material is located, a portion is collected and retained for further processing. The DNA screening section utilizes traditional detection methods to test sexual assault evidence for the presence of biological material and has developed a new Y chromosome screening method to more efficiently locate foreign male DNA on evidence within sexual assault kits. Y screening is more sensitive than traditional detection methods and allows for the detection of male DNA even in an abundance of female DNA. The method has also been automated, allowing for more thorough and quicker evaluation of sexual assault kits to identify those items which are suitable for DNA profiling and have the highest likelihood for success. Sample processing occurs in a biosafety cabinet, where the airflow is controlled to reduce the risk of sample contamination. Before DNA profiling can occur, the sample must be purified and the DNA isolated. Chemicals and heat are used to break open the cells, and then a robotic system is used to isolate and recover the DNA. The purified sample is then quantified to determine the approximate amount of DNA present. The process of amplification occurs next, during which the sample is amplified or copied at 27 specific locations along the DNA molecule. A robotic system prepares the DNA samples for quantification and amplification. It is driven by software-generated work lists, and samples are tracked by a barcode. Automation reduces the occurrence of human error and increases overall throughput, making processes more efficient and effective. The amplification or copying process produces millions of copies of the DNA molecule at the 27 specific locations tested and allows minuscule amounts of DNA to be analyzed. Subsequent processing on a genetic analyzer separates and detects the new copies. The resulting DNA profile is a visual representation of the DNA molecule at the 27 locations tested. Those areas are known to differ greatly between individuals and can be used to distinguish the DNA of one person from another. An evidentiary profile is compared to the profile of a known individual to include or eliminate the person as the source of the DNA at the crime scene. Evidentiary profiles can be entered into the Combined DNA Index System, or CODIS, to be searched against the almost 20 million profiles in the national database. The CODIS section populates the state's offender DNA database, which contains almost 390,000 offender profiles. Saliva is collected from qualified arrested and convicted individuals by law enforcement agencies and the Department of Corrections. The saliva is placed on a sample storage card, which prepares the sample for DNA profiling. A robotic system is used to collect a very small punch of the stain, which is amplified and processed on a genetic analyzer similarly to the other DNA disciplines. This overlap in processing techniques allows the disciplines to share laboratory space and equipment for increased efficiency. Offender profiles are uploaded to CODIS for daily searches. When a hit occurs between a crime scene profile and an offender profile, the offender's information is released to the case-working laboratory. A laboratory report is issued reporting the investigative information to the submitting agency. This new addition has benefited the Missouri State Highway Patrol and the Crime Laboratory Division by allowing the casework, screening, and CODIS sections to work side by side to produce DNA profiles and associate known individuals with unknown DNA profiles more efficiently, thus assisting the laboratory in managing the case backlog. This powerful investigative tool assists law enforcement in identifying and exonerating suspects, locating missing persons, and putting a name to unidentified human remains.